also he has been audited many of the international exposure he is having, like FDA, FDA USA, MHRA UK, TGA Australia, and Visa South Africa, as well as WHO. So today he is going to present a topic that is the processing of OSD pharmaceutical formulations and opportunities in OSD pharmaceuticals. So it's our privilege to have you as our speaker, sir. So now I welcome Dr. R. Kribakaran, sir, to take over this session. Welcome you, Honorable, sir.
Now I kindly welcome our chief guest, Mr. R. Kribakaran, sir, Manager Production, Adcock Ingram Limited, Bangalore, on the dais to give a guest lecture about the topic, Processing of OSD Pharmaceutical Formulation and Opportunities in OSD Pharmaceuticals. We welcome you, sir.
தனி ஒருவனுக்கு மருந்து இல்லை எனில் இந்த உலகமே அழிந்து விடுகிறது. So proud of pharmacy. We are all pharmacists. Also we are Indian pharmaceutical sector. We are part of the pharmaceutical sector. We have to proud because Indian pharmaceutical also known as pharmacy of world. India pharmaceutical exporting 2000 countries and territories, but US, UK, South Africa, Brazil, Belgium are top 5 destinations. India has large number of years of the membrane plant out of US. India supplying 80% of vaccine throughout world. India playing major role in pharmaceutical sector. It is producing around 80% of antiretroviral drug by India. India supplying 40% of generic drugs over US. India supplying 50% of generic drugs South Africa. India supplying 20% of all medicine to UK. We are all proud of pharmaceutical sector, pharmacy sector. We should be proud. You are all aware of that. I am here to explain processing of OSD formulation. It is easy for me because whatever I am going to explain the process and whatever I am going to describe technical words already knows. This is interactive section. If you are giving answer, I can complete within half an hour. Otherwise, it will be 1.5 hours, 2 hours. The question is 2 hours. Okay. You know pharmaceutical having what is the department is there? Any department? Can you tell me? R&D, quality assurance, QC, production. Yes. Whereas R&D, function of R&D, function of production, function of quality assurance, function of QC. Ask the question is easy, answer is better. Correct? Okay, I am going to explain everything in short form. You can understand because already you know the process, you read the process, easy to understood. That's what I am going. Only I am going to order of process. Already knows all whatever process he knows that already read. But I am going to otherwise I am explaining. I am going to take you and industry whatever we are processing the tablet, capsule, because this is OSD, especially OSD, that's what I am taking this tablet and capsule. Okay. Whenever we are making the tablet, making tablet is supposed to be formulation development has to do. You know that tablet having active ingredient, diluents, binder, lubrication, loud. Okay. Whenever doing the formulation development for comparability of active ingredient, they are developing and it will be the technology will be transferred to production by technology transfer department. There is an in between FD production technology transfer on department. They will transfer the technology. Then it will come to production. Production, for making production we need material. Material should be procured by purchase department. There is a purchase department. Whenever we are purchasing, where we have to purchase, that should be audited by us and that should be approved by us. Everything we have 3 approved vendors. Whenever we are getting short of one vendor, one supplier, then we are getting another supplier which is already approved. Without approval, we can't take from some other shop, some other suppliers because that should be validated. Validation, what is meaning of validation? When one thing we are doing, that should be validated. Whatever limits we are specifying, that limit should be there. Within limit we have to be done. That within one batch we can't validate it. We have to validate 3 batches. That should be, value should be maximum. Yes sir. I already told that this is interactive session. If you are not interacting, it will be goes more time. That's what we have to complete fast, then we will go. Because you know that process, that's what I am asking. If you are not knowing, then I will not be asked. Okay. Once validated, then that material will be purchased, then it is going to, once received material, that should be inspected as per our requirement. That once inspected, that should be sampled. That material, after sampling, it will go for testing. Test is completed, then it is on approval stage. We have one system, that is SAP system. Whenever we are creating the batch manufacturing record having what to be done, that instruction to be followed, whatever instruction to be followed, that should be there. Formula should be there. That formula given in the BMR, whatever materials we are receiving, that is not small quantity, that is bulk quantity. Based on batch manufacturing record, requirement will be dispensed. Means quantity should be dispensed accurately. When material dispensed, then it will pass to manufacturing area. That should be manufacturing. Having you know that there is a granulation stage. Granulation, what types of granulation? Dry granulation, wet granulation. Yes, good. This is what I need. Direct blending. Okay. Dry granulation. What is meant by dry granulation? Material should be and excipient should be blended. That should be compressed, pre-compressed. It means that name known as slugging. Okay, it should be slugged. Fine powders to large particles. That should be slugged. Then again it is going to milling. Cut into small pieces. Whatever record should be milled. Then it is taking lubrication. Then it will goes to QC testing. After get approval, it is going to compression. One more, direct blending. Some materials are having naturally compressibility nature. Then that should be blended with the lubrication material. That lubrication we are sending to QC for analysis. Once get approved, then it is taking for compression. Wet granulation. Wet granulation having so many processes. That I am going to explain that. When material is dispensed, there is a different material. Already I explained that different material. One is binder, one is dry mix material, one is lubrication. Okay. That material should be to be segregated. So, that material, whatever dry mix material, should be passed through shifter. You are aware of that shifter sieves. What is sieve? Sieve. Mesh. Murkla sieve button. Sallara. Okay. Why we are using sieve? We have to assign specified particle size of powder. Then one more, to remove foreign particle in the raw material. Once sieve, how do we measure sieve? Sieve, how do you measure? Force. How many force will be in the 1 centimeter square? 1 centimeter square. You have to draw 1 centimeter square, or one paper will be there. What is paper? Graph sheet. 1 square centimeter. You have to keep below the mesh. Then you are within 1 centimeter square. 1 centimeter square says once all 4 corners square. Correct? All 4 corners equal 1 centimeter. That should be counted. When counting how many pores are available in that is the mesh size. Whenever pore size is less, number of particle size will be more. Whenever pore size is more, then particle size will be less. You understood the principle? Yes? You are not aware about how to measure the mesh? Okay. Once material sieved, then we are taking to dry mixing. Between whatever binder that should be prepared. Binder having different binder. One is aqueous solution we are preparing. One more is non-aqueous. Whenever aqueous we are preparing, that should be used within 24 hours. Aqueous means water. Actually, water that we are using, that should be used within 24 hours, or otherwise partly lifetime. Microbial growth. When we are keeping material mixed with water, then there is a chances of microbial growth. If you are using non-aqueous, there is timeline for 72 hours. Because when using non-aqueous, it will not be available immediately. It will take time. That should be validated. Not 24 hours or 72 hours. Based on the product, based on the material nature, that will be different. The time will be worked, time will be validated according to the during development. When development doing whatever research doing, that according that they are fixing the timeline for keeping material. Once binder prepared, the dry mixing we are doing. What purpose of dry mixing we need? Dry mixing in the sense active ingredient, diluents, whatever excipients we are putting into mixer, then mixing. Why mixing is required? Anyway, we are going to add binder, then we are mixing. Why pre-mixing? That is dry mixing, why we need? For even mixing, we are adding active ingredient, excipient. That should be mixed properly. If it is not mixed, one side active material will be there, one side excipients will be there. Whatever we are taking to compression, that should not be having actual action, because one side having more drugs, that also problem. One side having less drugs, that also problem. This will not work. This will affect more. Then finally, we are getting serious. Once dry mixing completed, we are taking samples. That sample should be analyzed. That should be tested. We are taking sample different portion, different places, because we are checking different places whether that active ingredients, diluents, whatever other materials are mixed properly or not. Whenever we are telling mg, what is mg? Milligram. We are telling paracetamol 500 milligram. We can take directly paracetamol 500 milligram. We can measure in house. We can't measure. Whenever we are taking, that should be formulated. Why? With the other excipients. That is easy to take. Because if we are taking directly 500 milligram, it may affect directly affect our body, or it may not measured and weighed. Will not be. If we can't be tell this is I am measuring 500 milligram. That's what we are making the formulation. Once the dry mixing completed, whatever we prepared binder solution, we are adding to that. Then we are granulating. Once granulation completed, that it will be converted to tough mass, because previously that is fine powder. Now it is a tough mass that should be passed through wet milling, because that granulated materials is different size, bigger size granules. But based on the requirement, we fix the wet mill screen that should be passed, then it is taken to fluidized paint dryer for drying. Okay, now whatever I explained, vibrate that the sifting, what is the critical parameter in the sifting? What is the critical parameter? Seam. Whenever we are telling seam, what is the critical parameter? Critical parameters. The seam may be damage during processing. That wire, whatever wire, may contaminate our coat. Okay. Whenever they are asking seam, what is the seam? Critical parameter means seam integrity. Seam integrity, maybe that seems elaborated more. We can't get that specific particle size. Correct? Then we are taking to mixing. What is the critical parameters of mixing? What will affect? The speed of mixer agitator. That blade is there, no mixer. Already we are using no mixer in house also. Whenever we are keeping 1, 2, 3. When we are keeping 1, that milling will be different, that mixing will be different. When we are keeping 2, that will be different. Based on that, we have to be validated with the specific field. Whenever we are doing first, that should be followed until whenever we are doing that batch, that speed should be maintained, or otherwise there may be chances of failing. Batches failing, there is chances of improper mixing. That whenever doing wet mixing, we are calculating by ampere load. You have already engineering paper also. Correct? I am talking about electronics. That is engineering paper. When ampere load will be more? When ampere load will be more? When ampere load will be less? It means that the current, when current need more, I am adding dry mixing powder. I am starting the mixing. We need more current? No. Whenever I am filling full by mixie, I am starting the mixer, what will happen? We need some more current. Correct? No? When tough mass to be milled, that should be different. When light mass milling, that will be different. Okay. For that, we are checking whenever batch we are taking that current we are measuring. According to all 3 batches, continuous 3 batches we are monitoring. We are fixing these to this is a current. Because always manually we can't check whether this is granulated, this is the correct stage. But we can believe our machine. Machine will not tell false. Machine will not tell false. Because once we are validated, that should be followed. Then based on this current only, we are fixing that this is the granulated stage, very granulated stage. Then we are taking milling, I told milling. Milling time, what is the critical level? There is some different screen, different size of screens we are using. There is a different speed. When we are making maximum speed, that particle size will be fine. Whenever we are slow speed, that particle size will be more. There is chances of more or less. That also we have to be fixed range. This is the range that should be fixed. Once milling completed, we are taking to drying. Why drying need? Why need drying? Because already powders are in wet condition. That should be dry. If it is wet condition only we are keeping, what will happen? Again microfilter. Correct? Then that should not be taken to compression. Why? With the wet mass we can't compress. Correct? Then that should be dry. Then we have to maintain the loss on drying. What is loss on drying? Removal of water content. That whatever present in the granules, that should be validated and that should be fixed. Always we can't fix with the stringent limit. That should not be maintained. Every batches are a different person wise also there may be a chances of varying. That's what we have to fix with the elaborated limit. It is not should be stringent. That within that stage should be checked until packing, until final product. Or otherwise that also having gone. Once got dried, then we are taking now whatever dried granules not having the same particle size. We need particle size of the granules should be based on average weight of tablet. What is average weight of tablet? What is mass of tablet? I already told that paracetamol 500 mg being that not only 500 mg covers that having other excipients, that may be a chances of excipients plus lubrication 650 mg or 700 mg or 800 mg or 1000 mg also we can make. Whenever we are making 500 mg paracetamol, we are making 1000 mg tablet, what will happen? That is cost. We have to add more excipients. That should be also reduced. Whenever we are compressing, that should be a bigger pump size. That should be avoided. That should be reduced, but whenever that compressibility, for purpose of compressibility, we have to add that weight of powders, excipients. Then we are taking that whatever dried materials, whatever requirement of our granule size, based on that we have to select the multi-mill screen. What is screen? I already told You are seeing this? This is a multiple screen. That screen size measuring with the mm. We are measuring with the mm. That will be vary according to the particle size of the granules. Okay. Whenever we are pouring the powder, whatever that granules we are pouring, that will be milled, that will be passed through that screen. It means that what? Whatever we required size of granules will be coming out. It means that whatever we need, that granules will be coming out. Whatever bigger mass will be cut, and whatever requirement of required granule size will be coming out. That we are taking to blender. Then there we are adding lubrication material. Why we need lubrication material? Lubrication material for compressing of machine. Then there is a different lubrication material now. What is different lubrication material? Glide and machine lubricant. Different materials are that should be added into blender, that should be blended, that should be unloaded. After sampling, that should be analyzed. Once gas approved, then it is taken for compression. Then once compressed, tablet how it is compressed? By compressing force, by applying force, applying pressure to die cavity. That die cavity that comes will be based on our requirement. What shape, what size, what is the average weight of tablet? According to that, it will be varying. We are fixing that it is compressible. What is the compression parameters? Compression parameters. What should be checked during compression? Average weight. Average weight should be within the limit. Then thickness of tablet. Then hardness of tablet. Why we need hardness? During transportation, it should not be clear. Then we are doing disintegration test. Why disintegration test? Then variability. What is variability? Variability. During transportation, that should not be variable. That's what we are checking by 100 rotation or specific timeline. We are doing based on the product. Then if it is really having problem, that should be avoided by some backing material support, then that should be done. It is possible by making hardness and wear. Once compression is completed, that should be sampled. Sample how they collect during initial compression, middle of compression, end of the compression. That should be analyzed. Once get approved, then we are taking for, if it is uncoated tablet, we are taking to packing. If it is coated, then we are taking to, what is the purpose of coating? To mask, to enhance appearance, then to release stomach or intestine, whatever it may be, that sustained release. Some release purpose, we are doing the coating. Once coating is completed, then it is taken to inspection. During inspection, they will check tablet by appearance of tablet. Twin tablet, if you are aware of the twin tablet, during coating, may be chances of cause twin tablet. It means that while we are applying solution for coating, for film coating, by the time maybe chances of two tablets get stiff. That is twin tablet. There is peel off tablet. What is peel off tablet? That film whatever thing that may be peel off. During compression, there is capping tablet. What is capping tablet? The tablets are capped. Why it will happen? Because less moisture or granules was not proper. That's what that tablet got broken. Sticking tablet. Why causing sticking? Maybe excessive moisture. Maybe affecting sticking tablet. Okay. Then once coating and inspection completed, then we are taking to packing. Different types of packing. What is different types of packing? Container packing, blister packing, strip packing. Okay. Based on stability data, based on old time study, different packing material will be selected. Maybe this open exposure, this tablet will not be affected, this tablet will not be degraded, then we can pack with bottles. Because once bottle removed, then 100 of tablets, 2000 tablets, we are using 30 days or 40 days, whatever container we are taking, that should be used. It means that open tablet will be during storage, it will not be degraded. When blister packing, based on stability data only, that packing types of packing will be selected. Once packing is completed, then we are sampling and different stage during initial, middle, end. Once completed, we are taking and finished product as QC. Once got approved, then it is dispatched into wherever we are going to sell, that is going. There is some different regulatory different regulatory is there and other countries. What is regulatory? Every country has own regulation. That tablet facility, how should be manufactured, how should be followed, that regulation, whatever code of control they are framing us. It is very tough. Means US having that whatever regulation that frame, that is very tough to follow. Even I already initially told, India has largest number of US complaint plant in India. Because we are making the drug with less cost, more quality. That's what that other countries are buying from us. There is a different country I told, US, US FDA. They will inspect our facility. Once inspection completed, they will give approval. Then only we can send drugs to that country. Or otherwise, one tablet also it will not sell there. Even we can't sell, even that respective area person, if we want buying from us, they can't take it us. Because we have the approval, then they can take. Like that, TGA for Australia. Regulatory body, TGA for Australia. SAFRA for South Africa. SAFRA for South Africa. UK, MHRD. US FDA, what is US FDA? US Drug TGA, Therapeutic Goods Administration. What is GMP? Good manufacturing practice. What is the good manufacturing practice? Facility should be on proper. Why that the GMP is coming? Why when it is came GMP? What is the purpose GMP was introduced? In 1937, there is 1,000 deaths because of whatever manufactured tablet was not proper. Something that poisonous solvent was added and distributed. That 1,000 deaths, even more childs are because of that. That US Federation they are forced to follow the GMP. Where whatever companies are making product, that should be proved it is safe. Even that the introduced also in 1941, again same thing happened. Then that GMP was still stringent. They made stringent, then again there was something they derived extra control, then now it is all the countries following GMP system. What is CGMP? What is current? Present. Because whatever development, whatever new system we are implementing, that should be followed. We can't tell I am following GMP, but I am not in current status. I am in old status. That should not be tell. That's what the GMP was introduced. Whenever we are making the drugs, that should be followed current GMP. Understood? Whenever we are making the drugs, whenever we are selling the drugs, that should be 100% safe, 100% efficacy, 100% pure. That should be followed whenever we have the written procedure. We are following SOP. What is SOP? Standard operating procedure. Whenever we are writing that, that should be followed strictly. It should not be deviated. If it is deviated any procedure, what will happen? If we are deviating something, procedure is there, we are not following something, we are deviated, what will happen? There may be a chances of not working drugs, or maybe chances of side effect, or at last what? Maybe it is causes death. Correct? No, that should not be happen. That's what we have to follow. Something happened, that should be investigated properly. Simply we can't investigate, okay, this is what happened, okay, like corrective action, not like that. What is really happened? That should be investigated. Based on the root cause. What is root cause? We are investigating. Something root cause. You hear about root cause? Yes, what is root cause? Something happened, that should be checked. Then where is the happened? When it is happened, then we are identifying, no, that is the cause, root cause. Because of this was happened. Then what should be the action? If it is happened this, then that should be avoided. Whatever happened, that's what to do. According to root cause, we have to make one risk assessment. What is risk assessment? What is risk assessment? What we have to do? I know that how many steps I can go front. I have to know. I have to redefine. I am seeing you and I am stepping 4-5 steps, it is okay. Then 5th step, I will be followed. That should be pre-planned, pre-evaluated. That is on risk evaluation. Whenever doing every steps having risk, that should be avoided. Means of suspecting something. We have to suspect. We have to write more and more. This will happen. Then we have to find out the cause to avoid. What to be done to avoid? We should be more vigilant. Then only we can avoid the risk. Once risk is evaluated, we have to make an action plan for affecting gas. In May, I told everybody this sticking, during compression sticking came. What to do? Where it has happened? Then we have to investigate. Maybe chances of more moisture. Maybe chances of whatever punches used in the compression may be rough surface or not they may be cause sticking. When it is captive, there may be chances of, in compressive stage, there may be chances of more pressure. If it is applying more pressure, however, we will get crack. Whenever there is some no moisture, then there is some less binder, it will not be compressed. There will be a chances of breaking tablet. That's what happened. That should be avoided by corrective action. If corrective action done for current batch, what should be done in future batch? Whatever we done in the wrong procedure, that should be avoided. How we can avoid? Again, we have to make one procedure. How easily we can make? Okay, you do it that water. That should be done by training. Training will not be easily. Because we are reading, we are writing exam, even compressed, then coating. Capsule filling. Packaging. There is some blister bag, bottle bag. Finished goods. Finished goods already I have explained whatever. That drugs having purity, active ingredient that should be on specific limit. Now it is coming to opportunities. 10 areas, 10 different areas. Nowadays, that is not possible because readable. Whatever you have written something, it is not readable, then no use. It not meaning that it will not be tracked. That should be legible. If you are writing 3 or 8, that should be legible. Then C, contemporaneous. What is contemporaneous? Means that whenever I am doing activity, now I am taking one step, that should be documented. That should not be. Whenever I am doing activity, something I am writing by pencil, then after that I am copying, that is not correct. Whenever I am doing, that should be documented and present. That is the contemporaneous. Again, accuracy. Whatever you are doing, that should be accurately written. Accurate. Then, now newly introduced that ALCOA principle. Whenever you are joining in the pharma company, first they will take signs of that data integrity policy. Because we people always we will write something, then we will do letter, that this will be there. That promise letter will be signed, then only they will allow to work. Now, whoever the following the regulatory bodies companies, they are taking promise note. I am following these ALCOA principles. If it is anything deviation you are doing, you are not present, you are not in time, you are not in, maybe you are with tea break, by the time you are writing document, if you find out something, then why we need this many data? Why we need this many data? Something happened, that drug was went to market, it is distributed something goes wrong. How we can track? By document. Whenever we are taking document, then we will get to methods where is that wrong happened. Then we can find out easily for easy traceability. That document always, whatever manufacturing documents, we are keeping until once expired that plus 3 years we are keeping that documents. After that only we are destroying the documents. Why we have to keep after 3 years? That is also tracking, for the purpose of tracking. Okay. We have to follow strictly on procedure. We have to be online. We should be followed everything. Then it will happen. Because other countries, even you are all aware of that. During COVID time, other countries are struggling for the vaccines. But we are sufficient to vaccine, even we are supplying other countries. This and all happened because of we have n number of skilled labor centers. Apart from other countries, we have n number of skilled laborers. even we are making production skilled laborers, innovation skilled laborers are more in India. And also we are making the drugs by less cost, more quality, because we are applying on ethical things. Always Indian has more ethics compared to other countries. That's what they are believing as they are taking drugs. Most of the countries, around 2000 plus countries, we are exporting. Compared to other countries, this is the third largest producer of pharmaceutical. India has third largest producer. They are believing as they are getting drugs. Then FR&D, AR&D. Because initially, whenever formulation developed by FR&D, AR&D department. In that, one is this packing development. But our packing, materials selecting, then labeling. You know that pharmaceuticals is very important, labeling. Labeling is very important. If I know I am manufacturing some product, I know that if I am seeing color, because I am the company working, I know that this color was this tablet. You know that? You don't know. How you will come to know? Because whatever label telling, that you are trusting. So label should be always important. What are information available in the labels? Product name, active ingredient, shelf life, manufacturer name, manufacturing expiry, storage condition, full information, pharmacokinetic, then action of drugs, side effects, who will take, who will not to take. Every information should be available in the label. Labeling is very important. Without labeling, if it is moved product, then we have to raise the hand, this I don't know. Because we don't know what is the product. Without label means we can't conclude this is the drug. You can conclude. Because label only very important. And now without label, that should be destroyed. If it is missing label, that should be destroyed, that should not be used. Because we don't know when it is expiry, whether it is expired drugs or unexpired drugs, we don't know. That's what it should be clear that labeling should be used. Because our country, they cut the tablet one by one tablet, there is no information, then we are taking the drugs. But other countries not like that. They take us one full carton. Carton in the sense blisters and number of blisters available in one carton. But number of cartons goes to shipper. We are seeing nowadays bottle also cheaper than water bottles. Previously single bottle. Because easy to carry. That's what the shipper wrapping and sending. So we should be followed strictly procedure. Whenever you are coming in industry, that should be followed. You have to promise that the alcohol will be followed. Technology transfer. I told whenever FR&D completed, that technology will be transferred by that department. Then store or purchase. Material will be purchased, stored, dispensed by store department. Then production function. Production, manufacturing of drugs, manufacturing of tablets, packing of tablets. Then quality assurance. Every step they have to assure that the production has followed procedure or not. They have to assure. That's what quality assurance department function. Quality control. Whatever raw material we are receiving, whatever raw packing material we are receiving, that should be analyzed, tested, according to requirement, that should be approved by control. Means before production, they will plan monthly. This much product should be delivered according to the requirement of customer. Whenever customer they need, based on the need basis, based on the urgency, based on the availability of equipment, based on the availability of manpowers, then we have to plan. Then business development. Business development team will be getting business means orders from other countries. Business development. Then regulatory department. Regulatory department. We have a department regulatory that should be applying regulation, whatever regulatory bodies is there, no other countries, that should be applied. We having this many approval. You can take drug. We are manufacturing and giving. You can take like that. That is the function of regulatory department. Compliance team. Compliance. Once we are manufacturing completed, then that records, whatever we done records, that should be verified. That should be checked. There is some limit. Whether whatever we done, that within the limit, that should be checked. Then they will approve. Operational excellence. Operational excellence, nothing but production. Yes, and we have to use whenever equipment's capacity. If may chances of equipment will produce 1,000 tablets per minute, maybe our production operator will only manufacturing 100 tablets. That should be checked by operational excellence team. Where we are lacking, why we are not producing that equipment having capacity of 1,000 tablets. That done by operation excellence team. I told you, whenever old scenario, only one person monitoring the 10 areas or 5 areas. Nowadays, that regulatory bodies telling that single person should be there in single activity. There is a chances of missing if we are doing multiple activities. Whenever doing multiple activities, there is a chances of mistake. There is some chances of mistake. That should be avoided. One person, one activity. You can see that nowadays whatever industrial interview call letters coming, they are not one question, two question like that. 1,000, 100, 2,000, like that only we need for big companies. For pharma, they are having more opportunities. In industry, having more opportunities. You believe that engineering having not that much, but pharmacy having more opportunities. You can come and you can work without, because if you are completed, immediately you can get the job. That's what more opportunities there that you can utilize. Pharmacy having the more future. Thank you all. This is the time. This is the time to thank you, Dr. Principal. Our principal has forcefully asked me to give a lecturer because this is the session, you know that this is the pharmacology, especially
அந்த பாட்டு நேற்று 2 நாளாக ஸ்டேட்டஸ் வச்சுட்டு இருக்கிறேன். நான் உங்கள்கிட்ட எக்ஸ்பிளைன் பண்ணேன் இந்த பாட்டில் என்ன இருக்குது எங்கே கட்டல மதன் ப்ரோட்ட இந்த பாட்டு வச்சதுக்கு ஒன்று குறைந்து வந்து
Now I request Mrs. Neela Ma'am, Associate Professor, Department of Pharmacy Practice, Sri Adyani College of Pharmacy, to give an introduction about whole session speaker, Dr. A. Maleshwaran sir.
Good afternoon to everyone gathered here. It's my immense pleasure to introduce today's esteemed chief guest, Dr. A. Maleshwaran sir. Dr. A. Maleshwaran sir is currently working as a professor and head of the Department of Pharmacology in Kampala College of Pharmacy, Coimbatore. His academic journey begins from JK Madras College of Pharmacy as a B.Pharm graduate, followed by M.Pharm and doctorate program from Sri Ramakrishna Institute of Pharmacy. In 30 years of his experience as a teacher and researcher, he has to his credits 58 international and 4 national publications in peer-reviewed journals. He has the experience value of 15 and 658 citations in various research papers and authored 8 international publishing books. Dr. Maleshwaran's expertise extends beyond academia. He is a permanent member of various expert committees including the APTI, IPGA, and he is also served as a staff coordinator for the Scientific Service Committee, IPC, for the 3 consecutive years from 2013 to 2015. His dedication and achievements have earned him numerous awards. The Best Oral Presentation Award in the National Conference at JSS College of Pharmacy, Ooty, and 10th Pharmacy Teacher of the Year 2024 by APTI, Tamil Nadu. Ladies and gentlemen, please join me in extending a warm welcome to our distinguished chief guest, Dr. A. Maleshwaran sir. Sir, please welcome sir.
Now I kindly welcome our chief guest, Dr. A. Maleshwaran sir, Professor and
My second biggest challenge is the session. And you all guys are sitting after the lunch and that to the last speaker, right? And your mind starts to be there that when the particular speaker is going to complete the session, when I am going to receive my certificates. But put together, just keep aside my presentation. It's going to be an eye-opener for you that what are the alternative methods are available as of now. How you can use wisely the animals. Because each animal will have its own life, and when the life of animal is sacrificed, we have to give the justice to the animal. For this particular reason I have been used you. Even the animals are not having an ability to talk, but there is a humane care which we have to take care of animals, and that is the main motto of my presentation. So right now, what are the various animals are used in the research? In that particular matters, you people are already aware of it that mice and rats are going to be very commonly used animals in the research. What may be the reason? Any guess? Louder? So for a kind information, the mice and rats are so easily handled. Right? When I am asking you to do a research on chimpanzee, is it possible? Why I am asking you to go for a chimpanzee in the sense, whenever if you are looking for the structural similarity, the 99% of the structural similarity which is there for the chimpanzee to the humans. But for that we cannot go and conduct a study on chimpanzee. Then we have to look for the availability and the feasibility. So I have given here the statistics, right? That is in the USA, there is a top 10 organization. They are using the various animals, out of which the mice which plays a vital role, that is 83.6% of the research which is carried out by the mice. And the second highest is about fish. For a kind information, this is the statistics, is not from the India, it is from the US organization. So they have given the first preference for the mice and followed by the zebra fish and followed by the rats. Right? So this is the current scenario. So based on this, actually the CCSCA, you may be aware of it. So Committee for the Control and any guess? Actually CCSCA or CPCSCA? CPCSCA, right? CCSCA. But for a kind information, I got a crowd that they are aware of it that the CPCSCA has been changed into CCSCA in the last year, May month 24. What they have done, they just remove the purpose. The animals cannot be used for just purpose. So it is a Committee for Control and Supervision on Experimentation on Animals. So we have the CCSCA board. It will control the animals used for experiment purpose. And followed by, we may have an OECD guidelines. There is another one more guidelines which is available. They may frame the guidelines how to carry out the acute toxicity studies on animals. So these are the governing bodies which is available to control and do the research on the animals. And let me discuss my topic. That is, I have told you about the 5R concept. Right? So you people are aware of that is 3R concept, because so far, just 2 years back onwards, we have been so focusedly discussed about 3R concepts. That is called traditional 3R concept for alternative animal usage. Whether you have come across 3R concepts. What are those? Replacement, refinement, reduction. Whether the order is right? So I am going to change little different. That is, at first you have to look for replacement, then you have to look for reduction, and you have to look for refinement. So this is called 3R concept. That is otherwise called as a traditional 3R concept. But now, in this particular session, I am going to discuss about the 5R concepts. Whether I am going to put another 5 different R, no. All the first 3s are going to be the same. The 4th R, what I am going to give it to you is, it's about relevance. The 4th R concept is going to be the relevance. And the 5th one is robustness. So this is the 5 I am going to discuss with you all today. Right? So in that, each and every techniques we will be discussing in detail with various techniques. Right? Is this still? It's fine. Yeah. So it happens, right? So what you aware of replacement techniques? Replacement techniques for the idea. The first are replacement. Replacement is replacement. You have to replace. Whether we can replace the principal investigator, that is much better, right? Instead of doing one. So replacing is nothing but we have to replace the animals, right? So replacing of animals in the sense, what would be the technology we can go for? But nowadays it is possible to replace the animals. Without the animal, we can do n number of the research by using the various novel techniques. Thank you, sir. And yeah, absolutely. In between, I just want to point out one more very important information that actually you people are aware of. There is a various number of animals are there. That is rat and mouse, rabbits, pig, cow, horse, dog, and cat. So you can ask me a question, sir. Still, the dog is used for an experimental animal. Dog is used for an experimental animal. Use Pandroma? Yes, it has been used for cardiovascular diseases. Still, for the hypertension model, the dog is the only animal which you can go for the screening of the drugs. So, whenever the screening of the drugs for the anti-hypertensive drugs, you have to use dog. It is ethically approved. So, in a class, in a college, you use for a kind information for that, you have to get an approval for large animals. Right? So you have to get an ethical approval for to carry out the research on large animals. And even the cow and horse, still it is being used for vaccine developments. So like that I have been pointed out here. And as I told you, there is a various government bodies. That is CCSCA, what we have been discussed in detail. And one more is called AAA NAC, that is Association for Assessment and Accreditation of Laboratory Animal Care. And Organization for Economic Cooperation and Development, that is called OECD guidelines. Right? What is the purpose of to have all those organizations? So these are the organizations which give you the guidance to improve the animal care. Humane care has to be given to the animal. That is the major purpose of having all those committees. And for a kind information, nowadays you can conduct a study on humans very easily, but you cannot conduct a study on animals so easy. Right? So you have to face an IAEC, Institutional Ethical Committee, and for a kind information, Institutional Ethical Committee is not constructed only within the institution. The CCSCA nominate members, main nominee and link nominee, they will be there in the committee and they will ask you the n number of questions. Why you are using this animal? Whether the particular study has not conducted in any way. So these are the questions that the investigator has to give the answer for that. Then only you can get the approval for that. So that's why I am just stating the particular point that so conducting a study on humans is little easier when compared to an animal, right? So this is the 5R concept which I have very clearly explained to you. The first 3 is going to be the traditional, the next 2 is going to be the novel. And for a kind information, so I take a 5R concept here. In future, 12R concept already it is under proposal. Right? 12R, just imagine, double the fold. But what is the purpose? To reduce the animal usage, to increase the animal welfare. That is the major purpose of having increase in the number of hours. When we go for the replacement technique, as I told you, there is an n number of novel techniques are developed nowadays. Out of which the various techniques are called organoids, tissue engineering, and high throughput screening. So these are the novel technology which nowadays vigorously it is developing. So what is meant by organoid technique? Anyone can guess? And for a kind information, the next 15 minutes I am going to talk about very advanced information. Sir, part of the 15 minutes, very basic information. Right? The first R is going to be highly advanced information. In the sense, organoid. So you can develop an organ with a single tissue or cell, and that is what organoid. It is too simple to the doubt. How we are just going for the developing the cell culture. So similarly, you are going to develop an organ culture. So just imagine, this is a pictorial representation of brain organoid. You can develop a human brain by having an iPSC. What is the iPSC? It is an induced pluripotent stem cell. Right? Induced pluripotent stem cell. Within a one single stem cell, you can develop the entire brain organoid for your research. Right? If you want to screen any particular neurodegenerative diseases for any neurosubstance, which is possible by using a human brain organoid. Right? So here, the human brain organoid model, once you develop, so you can use for disease modeling purpose. You can screen the drug or chemical moiety, whatever the new chemical moiety which is available nowadays, that you can screen with the help of the human brain organoid. And you can reduce the animal model. Yeah, there is no need. I already having a brain humanoid. Why do I have to kill the animal? I have to isolate and brain and I have to carry out the spraying models. No need. Right? Directly you can carry out the study by using a human organoid and personalized medicine. This is one of the novel concepts nowadays. Because ancient days, right? Not even ancient, even nowadays, there is no personalized medicine. Out of this particular crowd, if anyone is getting a headache or fever, so what is the drug you are going to take? Paracetamol, Dolo, 650, right? Paracetamol 650 mg. Whether it is going to be the same for each and everyone, right? For your kind information, if a 60 kg of a person is going to take a 650 mg of paracetamol, and that is a normal dose, right? 30 kg or 40 kg a person, it is going to be the underdose. And 80 kg a person, it is need extra dose. But that amount of, that kind of personalized medicine, it is not available nowadays, and that is little difficult to do. The US countries and UK countries, now they are working on the personalized medicine. How can develop? A lot of strategies they are just putting on it. Right? But for normal, acute diseases, it is not possible. But just see to that, neurodegenerative diseases like Alzheimer's, Parkinson, or amyotrophic lateral sclerosis. For these diseases, there is a need to develop a personalized medicine, and that can be possible by using a human organoid models. Right? So the organoid models is not only for brain studies, it is also very vigorously working on cancer research. Right? So once again, the cancer patients, the metastatic cancer tissue will be separated out, and the tissue will develop into a cancer organoid, and the normal tissue will be separated out, and that will be developed as normal organoid. And for these two organoids, you can give the drugs, and you can look for the genetical modification and what about your therapeutical effect of the individual drugs. So this kind of the studies can be easily conducted under the organoid model. So what are the various applications of organoids? That is revolutionizing the drug discovery workflow and evaluate the immunotherapies and explore the genetic disease drivers, and also you can get into the in vitro to in vivo translation and identify the predictive biomarkers, and finally you can track the tumor cells. So these are the various applications of organoid models. Right? The next comes tissue engineering. So far what I have told you, I have told you about the cell. Right? Or otherwise, if you are taking a simple one tissue or one cell from an organoid, you can develop the pluripotent stem cell. Whereas here you are going to engineer the tissue. The entire tissue can engineer, and for that what you require is you need a pluripotent stem cell, and there is a need of an tissue architectures. So tissue architectures, which is so helpful to develop and to make into a tissue engineering concepts. Right? And with the help of the tissue engineering concepts, what you can do is you can develop the entire tissue architecture. Right? And that in the tissue you can do, or you can give the drugs, and you can look for the changes in the genetic modification, and finally you can come to a conclusion. The next one, what I told you is, under this tissue engineering, the novel technology right now is available is 3D printer. Right? So 3D printer, it is, you may come across in case of the IT technology, but that is also used in our life sciences. So with this 3D technology, what you can do is, so you can track the trachea, heart walls, skin, as well as the bone, the depth of the fracture, how it has been happened. So these many informations can be evaluated by using a 3D printer technology. The next comes the applications of tissue engineering. As I told you, from the tissue, that is biomaterials, biophysics and transportation science, this all can be covered under the tissue engineering concept. Next comes high throughput screening. I think most of you may come across the molecular modeling, right? That is called high throughput screening. In the sense, whenever I think earlier, just 10 years ago, there is not a very big development in the software which is available for drug discovery. So there is no need that if you are having 100 structures, there is no need to get into all the 100 structures and to synthesize and to screen onto the adenine. There is no need. You can select only 5 drugs based on their binding energy, right, towards the target. And for the 5 drugs, you can put into an in vitro studies, not in vivo. And the best compound what you are getting, that one compound or 2 compound you can get into an adenine. Just imagine 100 compounds, you are just cleaning into just 1 or 2 compounds. So that much level of attachment is available under the high throughput screening. Still I am getting a lot of challenges apart from YouTube. So let me go for the in silico models. Right. As I told you, there is a molecular modeling, artificial intelligence, and physiological based pharmacokinetic model, that is PBBK. So these are the 3 different models which is available under in silico model. So molecular modeling, which you have to develop the structures and you have to organize it and you have to develop a drug molecule. So here, what are the various applications for the molecular modeling? Where you can identify the drugs and targets, as well as you can screen the drugs, and you can develop the virtual screening methods and admit properties you can study, that is your pharmacokinetic, as well as toxicity parameters can be evaluated by using a various softwares like NADU designer which is available, which can easily predict your pharmacokinetic profiles. The next comes to the global scenario of molecular modeling. So now, the molecular modeling which bears a huge market value, that is around 67.56%, which has been covered under the life sciences, which is approximately 140.4 million US dollar market is available for molecular modeling concepts. The next comes the artificial intelligence. So artificial intelligence is also getting involved in our pharmacy sectors too. So we cannot defend or we cannot predict the artificial intelligence. It's nowadays entering in each and every local government. So this artificial intelligence, which is used in assay and developing the synthesis and designing the molecule. This all the 3 phases, the artificial intelligence plays a crucial role, especially in case of diagnosis. The fractures can be diagnosed and diabetic nephropathy can check about based on the organ volume how much amount of the drug which is present on the organ. So that kind of information you can bring it down, and that is going to be your absorption, distribution, metabolism, excretion information. Right. The next comes a very vital technology, organ on a chip. Right. So organ on a chip in the sense this all those PBBK model. Right. So that can be easily kept a chip inside of your body. So just imagine. I think this is not going to be the very toughest for you, right? But easily you can understand that nowadays the various films that is it is coming at that a person can be inserted with the chip to know about the activities of the particular person. So do similarly here we can assess what the drug is putting its effects, what kind of the release of the drugs that can be measured by keeping a chip inside the body parts. So what are the different that is chips are available in the sense? Just imagine. So blood-brain barrier, that is in the brain, DPP chip, as well as lung on chip, and liver on chip, and kidney on chip. So these all the organs where you can place a chip and you can monitor the physiological function as well as the pharmacological effect of the drugs. So
which gives you the exact picture about how to check the genetic variations. So what are the kind of genetic variations which can be carried out by using a CRISPR cell in a sense? For a kind information, this CRISPR modified cells also can be produced from induced pluripotent stem cell. That the thing is, here, what are the possible methods? That is, you can go for knock-in, knock-out. You may be come across knockout model, right? I think knockout model of the rat or mice, but here they are not going for animal, they are doing on the cell. So where you can knock out a particular amount of the organs or particular amount of the tissue, and thereby you can conduct a study and come to a conclusion. That is called CRISPR modified cells, right? Where you can, here it is not only for knockout, even if you want a particular organ that you can insert. So that is called knock-in. So that is also possible, and even these CRISPR models are used for diagnostic purpose and to screen the drugs. So this all it is possible by using the CRISPR modified cell technology. Next, very quite interesting topic, zebrafish. Right? Nowadays it is very vigorously developing, but I am going to ask you a question: whether the zebrafish required ethical clearance or no? Yes or no? Then why I am keeping here in alternative models? Right? Zebrafish, if it is required an ethical clearance, then why I have kept? There is a two reason. For a kind information, before last year, that is 2022 end December, not even December. So 2023 of March, up to the study you can, because it comes under the vertebrate model. But once again, in the last year, they had put into the CCSCA, they had get into the picture. So they asked that even if you want to frame a study on zebrafish, you have to get an ethical clearance. Then okay, sir, why you have to do this? But for a kind information, if you want to get an animal clearance for rat and mice, it is difficult when compared with the zebrafish. Because the zebrafish reproduction rate is much higher. Actually speaking, the CCSCA is giving you the guidance that you have to use rat and mice, not in larger animal. What is the reason behind that? Because rat and mice are going to the smaller animal, reproduction rate is higher, so automatically you can use those animal effectively. That is the basic reason behind it. By keeping the same in the institutional ethical committee, if you are presenting a research protocol on zebrafish, you can easily get the clearance. That's why I have kept this as one of the alternative tool or alternative model. So here I just want to add one more information that the zebrafish having one peculiar character. Anyone knows? Anyone knows? I think I have given you the list of diseases. So obesity, atherosclerosis, diabetic retinopathy, stroke, arrhythmia, everything I have given. But one particular peculiar thing is there for zebrafish. Any guess? What is the cardiovascular specification? For a kind information, as part of heart is better than zebrafish, the zebrafish will regenerate the entire heart. Just imagine. So zebrafish should have heart and the heart, the remaining part will regenerate. How your nails are, whenever you are cutting out, automatically it is regenerating. Similarly for zebrafish. Right? And now the majority of researchers in US, UK, everyone is working on it. So which fiber is responsible? What is the pathway? How it is regenerating the cardiac fiber? If they are able to get it, then there is no need of heart transplantation. They will simply develop the heart regeneration. So still n number of research is going on, and that's the one particular point I had given you as a one more pictorial representation. That is, this regeneration is due to the RAS and MAP kinase pathway. It has been identified, but which fiber is responsible to develop the heart, it is still another question. So you all people are having a humorous chance to get that particular information. Even if you are having an idea or if you want to do it on the research, you can very well do, and you can identify that particular pathway. Then it is one of the wonderful and wonder in this particular research, cardiovascular research, right? So now, I think so far I have given you the enormous information. I don't know how much it has been understood for you, but basically I just want to give you a small recap. That is organoid methodology. So developing the entire organ from a cell, and CRISPR cell modified technology, right? And then computational methods, that is molecular modeling approach, and high throughput screening methods, right? So these are the various replacement methods are available where instead of animals you can use these methods. Right? What is the purpose, benefits, Abhimadina? The ethical alignment with reducing the animal suffering, and it is going to be the more relevant to the human biology. Right? That is one of the major purpose. And one more is about faster, cheaper and scalable. Right? So these are the various applications are importance for replacement technique. And let me go for the reduction techniques. So how you can reduce the number of animals? So here I am not going to go you very simple. You have to use the better statistical tool. So initially for each group 12 animals, 18 animals have been used for the study. But nowadays it is only 6 animals. And that is because of the ANOVA. What is ANOVA? Analysis of variance. It's the biostatistical tool. And now I am going to give you another one more example, that is power analysis. That is 2 to the power of 10. So 2 power 1 is 2 animals is enough for a group. So now that analysis also get into the picture. So what is needed is you should need developing a biostatistical knowledge. That is the myth. The pharma student they doesn't aware of biostatistics. Right? But you people have to create an interest because it is a part of research. So you have to develop a better biostatistical knowledge to reduce the number of animals. And one more is about the shared data and resources. Right? Majority of the time what we may be doing is we are unnecessarily wasting our energy. Right? For already known information. For example, if you want to study an anti-inflammatory activity of a particular drug or particular plant, the same work has been already done by somebody else and it has been reported in a scientific ground. Then what is the purpose of redoing the same work? Right? It is a repetition of work. So, which one is missing? NMSL, we are not knowing about the information. That's why what is my sincere request is you have to go for better review and meta-analysis before you are starting your work. In your work, you have to go for the complete thorough analysis. For this particular plan, already the work has been carried out or not? And for this particular plan, what is the novelty in my research? If you understand this particular way, there is no need to repeat the research and you can save the resources. Unnecessary money waste and your time, everything can be prevented. Right? So multi-endpoint studies. If you are doing a study on neurodegenerative disease, for example, I want to do a study on Alzheimer. Okay. What about the other organs? Right? If I wanted to do only one particular study and my study is going to be the monopoly, automatically the animal life, it is going to be wasted. So try to maximize the benefits. Is it possible to do any other kind of study? So multi-endpoint studies, multiple outcomes if it is possible. Then you have to frame a such kind of research. That way you can reduce the number of animals and you can reduce the unnecessary usage of the animals. Then next comes whenever there is a need of animals, we have to use. If there is no need to use. Because that is the core concept of CCSE. Right? For example, as I told you, molecular modeling, autodoping, if you are using 100 compounds and if you are converting into 5, there is no need to do the research on the remaining 93 compounds. And just imagine it's not 93 compounds, you are just saying 93 groups. And each group consists of 6 animals, 95 into 6. That much animals you are just saving or reducing for the betterment. Right? So whenever it is necessary, that time alone you have to use the animal. So this is already I told you should know and better statistical knowledge. So that will control the experimental noise. They will the noise it will be reduced. The next systematic review and meta-analysis before starting your work. Complete thorough analysis about the research work and the research question and the output. Everything has to be analyzed. Then thereafter you have to start your work. So these are the benefits of reduction. That is, the reduction will improve the ethical improvement. For your kind information, nowadays, I think I am one of the member of institutional animal ethical committee. Where whenever the proposal comes, what is the first point we may give it to the people you know? Remove the normal control. Remove the negative control. Right? What may be the reason? Already n number of studies for that particular disease, the normal control has been studied. So normal control is just an water and you are going to give an animal surviving for your study period. And after that you are going to kill. What is the logic in it? Remove the 6 animals. So this is one of the measures. So before I am just going to say that remove 6 animals, your proposal which have And that is my suggestion too. And next one is about cost effectiveness. When you are reducing the animal, the cost of the animal is going to be beneficial for you, as well as unnecessary waste of time can be minimized. So enhance the reproducibility and reliability of the results if you are going for the better planning. The next comes refinement, the third principle. It's the 5 principle, now only third principle, right? Another 2 more. Don't worry, I will make it so simple. So refinement, which will minimize the pain and suffering to the animal, right? The refinement techniques will reduce the pain and suffering to the animal. And what way we can do, or we can reduce the pain and suffering to the animal? By giving them better animal house and husbandry conditions and environmental conditions. So if you are giving the better environmental condition, the animal will reduce its pain. That is a simple logic. Okay. Just imagine. I think for we people has to accommodate, fan is more than sufficient, right? But if you want to store an animal, you have to, that means the animal house or the room should be air conditioned. Right? The animal house should provide with an humidifier. The person, those who are entering into the particular room, they should be free from microorganisms, well sterilized. So if you want to enter into the animal house, it should be a laminar airflow under the condition you have to enter. And you should wear a face mask, head mask, so everything. And you cannot put all the 100 animals in one particular cage. Right? So you have to space only the 6 animals per cage. It is a norm. What is the reason? If you are keeping large number of animals, it will change the behavior of the animal. When the behavior is changed, your results are going to be different. So, proper housing, right, and training of animals will minimize the stress. For example, if you want to do a study on Alzheimer's disease and you want to assess the behavioral characteristic of an animal, the methodology what you are going to look for is about just get an imagination of Maury's waterways, right? So if you are taking an animal, just put it into the water and ask to swim, it won't. Already the animal, whenever if you are taking out, it will be there in stressful condition. And if you are putting it into the water, just imagine, right? For a kind information, without you all people, that means without a program structure, if a program is running, so what is your attitude, the same is going to be for the animal. Right? But already it has been informed you that this is the last speaker session. So now you will be having some kind of reduced stress. Just imagine, no one has been given you the program schedule and asking to sit prolonged period of time. It is not comfortable. That the same thing is going to happen for the animals too. Right? Sir, what is going to happen? The ethical care and humane care. So if you are handling an animal, the animal has to behave like it is free from the stress. So that is the major concept which comes under the refinement technique. So refinement, when you handle the animal has to feel that it is safe. But actually students what will do now? So they won't have hold the animal like this. So they will put it on the tail, right? And they will throw it away. So end number of the pharmacology labs you may see like that. And actually speaking, the PCA also it is supporting you. Because now they have been totally removing the animals from your syllabus. So you doesn't know how to handle the animal. But see to that, this is one pictorial representation. The animal is so calm, so comfortable that need to be maintained to the animal. So what is the need for refinement? That is the last one particular point. Just look into this. This leads to the variation in experimental results which impacts both the reliability and repeatability. So first time if you are putting an animal into the water without training, automatically the second time you cannot expect the same results. So the proper training and proper refinement technology which will give it to you the reliable and repeatable results. Fourth concept, relevance. When the animal has the chance to talk, right? When it is getting a chance to talk, what is going to say? Arey yaar, 92% of the drugs tested on me is fine. 92% of the drugs which is tested on me is failed. Why you are unnecessarily clinging to me? Right? This is the question is going to ask by an animal. Each and everyone, just imagine. Right? I think for a kind information, I am going to reveal another one more information here. Just listen. For my PhD, I think I had around 126 animals I had killed. like this. But see to that there is 92% of the drugs it's conducted on the animals, it is getting failed. What is the major reason? The reason behind is the relevant research is not taking place. The irrelevancy which lead to the failure. So you have to look for the relevancy of the research. That the point is, whenever if your experiments are performed, that should be scientifically appropriate and that should be applicable to the human biology. For a kind information, all the animals cannot be used for all the research. I have already told you, vaccine research. You cannot take a mice and you cannot carry out the research. Because the biological relationship is different. And automatically if you are getting a research, it won't be a reliable one. So the proper reliable results will come to you whenever if you are selecting your animal species. So just imagine here I have given the comparison of human brain and an animal brain. So human brains are going to be the larger and more complex. When we are looking into an animal brain, it is going to be the small size and the complexity is going to be less. And highly developed and having a complex function, here it is going to be the simple function and less developed. So this is going to be a major difference between animal brain and human brain. Now I have given you the structural similarity. In my first discussion I have told you that animals and humans, the chimpanzee which is going to have a 99% of human similarity. Okay sir, Madheshwaran sir accept 99% of similarity. Hereafter whatever the research I will do it on chimpanzee. It's not possible. Right. So see to that the availability, reproduction rate and approval is everything plays a vital role. By keeping up those information, the rat it is going to have, there is the mice is going to have 81%, followed by I think the rats are going to have 78% of structural similarity. As I told you, zebrafish, there is regeneration of heart, it is having 55% alone. But even those 55% for cardiovascular research, still there is a miracle is going on the zebrafish. Because once the concept is identified, then there is no need of the heart transplantation, as I told you earlier. Then next comes the last principle. I think most of the faces are getting smiled. Right? Robustness. So robustness, it is nothing but reproducible, reliable and meaningful. Whatever the conclusion you have, it should be reproducible, it should be reliable and it should be meaningful. So what is the meaning of reproducible and replicable? That is the another one more technology. That is terminology. That is reproducibility and replicability. These two you should know about it. Because then only you can know about the robustness concepts. That is, reproducibility means you have to use the same data for the same conditions. Right? That the thing is the methods, data analysis, everything is going to be the same and you are going to repeat it the same and you are going to get the same results. That is called reproducibility. Same method, repeated open rate. That is called reproducibility. Whereas replicability means even if you are going for the independent experimental conditions. Now, any method how to get the same result. For a kind of information, Alzheimer activity will give, right? For Alzheimer activity, there is the n number of mechanisms are there. Cholinesterase pathway, tau protein pathway, phosphorylation. For any methods, a drug is working on it and finally give you the information that it has an anti-Alzheimer potential, right? And that is called replicability. So your study should show reproducibility and replicability. That is very, very important, right? So these are the various 5 concepts. That is replacement, reduction, refinement, relevance, robustness. So by putting this all the 5 concepts, you can rationalize the animal usage and that the animal needs. And so far, what I have kept here the presentation is, stop teasing an animals. Each animal has its life, and if the animal life is sacrificed, it should be justified with the various reasons. With that, I just want to conclude my presentation. Thank you all for your patient listening. Opportunity to thank Abhirami Management and the Principal of Abhirami College of Pharmacy, Dr. Jivanandan sir, and Organizing Secretary, Sir Dr. Srinivasan sir, and all of you for giving me this opportunity. Thank you all. Finally, before concluding, if there is any question, questions. That is two things. One is everything you understood, there won't be a question. If nothing is understood, there won't be a question. Right? It's up to you. I believe that, yeah, there is one question.
Sir, apart from the higher concept which you have explained today, I need to ask one important concept which you have explained under a particular topic, which is personalized or customized therapy system. Don't you think this is a reverse staging, or that we have used the recombinant medications a lot of years ago, which we are decades? And what do you think is the very significance of this particular personalized therapy system?
Personalized medicine. So see to that, very good question. Personalized medicine. What is the merits and demerits? The merits, I can say that this is right now it is needed, because whenever if you are going for the personalized medicine, the name itself is very clear. What you need, that I have to deliver. That is very, very important. But what is not possible is the Indian population. Right? The Indian population, for each and everyone, if I am making the personalized, and for that much amount of pharmacists so far it is not available. Right? That is the need. And for, I think, for the chronic diseases, sure there is a development of personalized medicine, and still it is working on it. For a kind information, for the cancer, you may be having like the colon cancer, vincristine and vinblastine. Right? Whenever the drug called vincristine and vinblastine is given, they will be looking for the body surface area of the individual, and they will personalize that particular medicine, they will be delivering. It is not unit dosage. So like that, for each and every diseases, if you are capable to go for the personalized medicine, it is very happy. But practically, it is little difficult as of now. Maybe the scenario change, the artificial intelligence are getting involved, then it is possible. It is not impossible, but now it is not possible. Right? Thank you.
So then what would be the effect of inaccurate dosage?
What will be the
effect of inaccurate dose in the surgical system?
No, inadequate, as I told you that if the dose is not sufficient, you will get a less pharmacological action. If the dose is higher, you will get a toxic effects. That is the major, right? So you need an exact dose. So that is called personalized medicine. The side effect is same. If you are under that dose, you won't get the pharmacological action at all.
Thanks.
Yeah. Shall I write off? Yeah, thank you all.
Thank you, sir, for your informative session. I hope the gathering gained the knowledge about alternatives to animals used in research for your concern. I request the dignitaries to sit on the rise. I request the dignitaries to sit on the rise. I request Dr. B. Srinivasan sir to be seated on the rise.
and memento to the participants. Now I would like to call upon the evaluator for e-poster presentation on emerging trends and challenges in drug safety, discovery and development, organized by Department of Pharmacology and Pharmacy Practice, DR.M.G.RMEDICAL College of Pharmacy. First, G. Sumitra, Professor, Erode College of Pharmacy. Please be on stage, ma'am. I would like to call upon the dignitaries on the rise to distribute the memento and certificates to the participants of today's event. So first, United College of Pharmacy. I request the faculty members of each college along with participants to receive the memento and participation certificate. So first college, United College of Pharmacy. I request the faculty and any students from that college to receive the memento and participate certificate. United College of Pharmacy.
தள்ளி வாங்க.
Next, Eton College of Pharmacy. I request the faculty member and the students to receive the memento and the certificate. SMS College of Pharmacy. Karpama College of Pharmacy. Karpama Academy of Higher Education. United College of Pharmacy.
Ready? Okay.
அப்படியே இருங்க.
Thank you.
I would like to call upon the dignitaries on the rise to distribute the participant certificate for the e-poster presentation. So the first participant, Pooja from RTS College of Pharmacy. Participant certificate for e-poster
Sir, okay, sir.
Janisha from PPG College of Pharmacy. Pratibha from PPG College of Pharmacy. KM Nisha from PPG College of Pharmacy. Rameshwari, Kaleshwari, Jayabharati, Keertana, Jeffrey, Jeevita from PPG College of Pharmacy.
Bro. Hello, bro. Hello, bro. Ready, kemana? Depan ya.
Pradeep from Gaham Academy of Higher Education. Jawahar from Gaham Academy of Higher Education.
Okay. Sir, okay.
Pushpendra Maharaj from JKM College of Pharmacy. Haribharati. Jiva. The students can come and collect the participant certificate. Veronica from EOD College of Pharmacy. Amitavarshini from EOD College of Pharmacy. Thank you, dignitaries. Gratitude is not only the greatest of rituals, but the parent of all others. Now I request Dr. P. Srinivasan sir, HOD and Professor, Department of Pharmacology, to deliver word of thanks.
So, as we come to this end of this seminar, so I would like to take a moment to express my heartfelt gratitude to each and every one of you making this event such a resounding success. So, I thank our respected chairman sir, Dr. P. Periyasamy sir, and also our board of directors, Devi madam, and Senthil Kumar sir, Umma Devi madam, Balakrishnan sir, Sushilita madam. So, even though in their absence, so not only they are supporting in this seminar, so they are providing wonderful infrastructure and animal facility to our institution. So, thanks to the management. So, they provide great support to organize this event. So, thanks to our Dean, Dr. Jayabharati madam, so for his constant support for this event. So, I really thanks our young and energetic principal sir, Dr. S. Jeevanathan sir, for providing great support on this one-day continuous education program. So, on the topic emerging trends and the challenges in drug safety, discovery and development. So, next I would like to thank our esteemed speakers, Dr. Gavani sir, and the Dr. Garan sir, and the Mr. Kirvaran sir, and the Dr. Madheswaran sir for sharing their valuable insights and expertise with us. So, your presentations are we are truly enlightening, and we are grateful for this time. The effort you take to prepare and deliver to the students is very appreciable. So, to our organizing committee and our P.R.M.G.RMEDICAL Pharmacy staffs, non-teaching and teaching faculty, I extend my grateful for tireless efforts in planning and executing this event. So, even though we have a national pharmacy celebration which is conducted from last week, so we have So I thanks recently every teaching and not teaching and all HODs. So your dedication and attention to the detail were evident throughout, and we are grateful for your hard work. So especially I thanks Google Park and Dr. Karan and the Sanas and the other teaching and non-teaching members. So finally, our fellow delegates from RBS College of Pharmaceutical Science, RBS Performing College, PPG College, United College of Pharmacy, and the Karpanam Academy of Higher Education, Karpanam College of Pharmacy, the Europe College of Pharmacy, JK Nirala College of Pharmacy, and Nanda College of Pharmacy. So that's the college of Chart. Okay, so I thank you for active participation and engagement through our session. So your questions and the comments and discussions were thoughtful, probing and enriching. We are grateful for your contribution to the seminar discourse. So in closing, I would like to say this seminar session having truly enriching experience for all of us. So especially I think all the sessions are very useful. From production, various departments, production, pharmacovigilance, and about the animal experimentation, and the new education policy about the stored by our companies. So I think all the sessions are very useful. So we have learned from each other, share our knowledge and expertise. So thank you once again, and I wish you all the best for your future endeavors. Thanks. So our academic students also. Thank you, Amanda.
Thank you so much for your speech. Once again, thanks everyone for your valuable